Hello. I'll start just by reminding a little bit what we saw yesterday. We saw that population, the minimal model for population growth is exponential growth, and this happens when this growth is proportional to the abundance or the density of the population. In this case, if we have that the growth is a constant multiplied by the abundance, then the solution for the system has an exponential function. One of you after the class asked me, there was some confusion about what will be exponential growth or super exponential or infra-exponential, sub-exponential growth. So if you find models in which this x, instead of having an exponent of 1, has an exponent greater than 1, you will have a supra-exponential growth. And with an exponent here on x lower than 1, then you have sub-exponential growth. It goes slower in growing than it will do with an exponential. Then we saw that populations at some point, when conditions are favorable, are going to grow in a way that we can capture with exponential growth, but this cannot last forever because this will continue growing to infinity, and we know that populations cannot grow forever. So when we think about the saturation of growth, the most simple thing we can think is that individuals from the population are competing with themselves, and we can see that in a linear way. So basically, now we add this term that captures the competition with the individuals from the same species, and this constant k is the carrying capacity of the system. When x reaches this k, then this term will be 0, and then growth is stopped. Okay. If we think about the per capita growth rate, which is for an individual or for a unit of the abundance, how fast is the growth, then we can always divide the total growth rate by the abundance of the population, and we will have here we see that there is a linear dependence on the saturation of growth under the logistic growth model. Okay. Then we know that this is not the only way that we can capture the saturation of populations. It could come in different forms as well. This is the most simple one. And some of them will just have an exponent on x. And the literature can be a little bit confusing because they call different models in different ways. But for the purpose of the form, we see that if an exponent on x here will be lower than 1, we can consider this as a sublinear saturation of the growth. Okay, but for now, we just continue with the logistic growth to move on. And what we saw that for logistic growth, we have this linear decay of the per capita growth rate of the population. All of this was thinking about the saturation of the growth rate, but actually this saturation comes from competition with the peers, with the individuals from the same species. But this is not always the only way in which individuals from a population can interact. For example, in different situations, they can cooperate. And this is the first thing that we will see today in class. And later on, we will move from populations of individuals that are identical to interactions between different species. So we know that populations that don't exist alone in nature, but we always find it in competition or cooperation in interaction with other kinds of species. And we will work on one of the most simple or canonical models for the competition between species, and we will start with populations or communities of only 2 species. So the minimal model we can think about. And finally, we will see how we can use this model to predict how these communities can respond to different perturbations. So populations can cooperate for growth in different ways. And one way that is relevant for microbes, for example, is when they have to digest a complex resource that cannot be imported directly through the membrane of the cell. So this happens, for example, for yeast populations. We have that when they are growing in sucrose, sucrose is a complex carbohydrate, and they cannot import it directly. So what yeast cells do is to secrete some enzymes, and these enzymes can digest the sucrose into two simple sugars. So here we have, sucrose. And what the enzyme that each cell is secreting does is to digest it into two simple carbon sources that are glucose and fructose. So as they are digesting it, there will be an increasing concentration of glucose and fructose in the system, and these are two carbon sources that they can import, and then they can use it for growth. Okay, so we can see then how this might be, or this is a cooperative process, because if we only have a few cells, or let's say one cell in the environment, there is only a limited amount of enzyme that can be released, and then after digestion, these two simple carbon sources can diffuse in the system. So we need we rely on the chances that one of these carbon sources has to be absorbed by the cell. If we have more cells in the system, we will have more of the enzyme, and this enzyme will be digesting the sucrose more effectively. So this will increase the growth rate of the population of the cells in this medium. So one way to model this is to try to capture all of these reactions that are happening in the system, and this can be done and has been done for the particular case of yeast, but there are many other processes that lead to a similar dynamics. And the most simple model we can have for just the population of cells, perhaps, is to consider the logistic growth that we had before, and add one term that captures now a quadratic form of the growth. So if we have these, we see that when the population is small, what we have is that this has this from here. And if we think about the per capita growth rate for this model, what we have now is R. So this per capita growth rate, now it's a quadratic form on X. It crosses 0 at K, same as for logistic growth, but then it also crosses 0 at C, at XC, sorry. It's a quadratic form, so this does something like this, which we see here that it introduces a minimal threshold in the population for it to survive and start growing. If we are above this threshold, the per capita growth rate is positive. It means that the population will grow, and as before, for the logistic model, it will reach carrying capacity. If we are above the carrying capacity, then it will move here. However, if we are here, the growth rate is negative, and this population will now go to extinction. So we have now one unstable point at a threshold for the population, which is called the Ali threshold, because this model for cooperative growth is also was originally modeled by Ali, and we also call this the Ali effect. Okay, very good. Now, yesterday we were also considering what will happen if this population is being harvested, and we model this harvesting, which could be hunting or any other kind of predation, by just adding a constant rate of death. And when we have this in the equation, we can think about this as adding a constant death rate that has to be subtracted from the growth rate, that the per capita growth rate that we had before. So in this case, when we analyze how this X star depends on the added death rate, we have something like this. It's K, and it goes down until the death rate is equal to the growth rate. In here, if we add now, sorry, this was. If we have now this term, what we have is that the population, this stable abundance, as we have the growth rate, is going to move to this parabola. And then once we increase the death rate above a critical threshold, then the population will start suddenly going down. Okay. So here above the critical threshold, the population reaches the stable state. There is an unstable state that is also changing. And at the moment in which we add a death rate that is exactly at the critical point, then the population will die and it will not be able to recover. Okay, so and we can see the qualitative difference here because as we add predation or hunting in a population that grows logistically, then we will approach smoothly the point of extinction. So if you stop hunting, it will be able to recover. You can see how it's as you are predating, you are approaching zero. So at some point you can react and lower the death rate, and the population will recover. But here we have what is called a fold bifurcation, and it can lead to a catastrophic shift in the abundance, the stable abundance of the population. So here if we look at the stable abundance of the population, is relatively high. So we might not predict that it's going to collapse only if we predate a little bit more. But if we do it, then the population will rapidly go to extinction. And even if we remove that death rate, the population will already not be able to come back to its previous abundance. Okay, is this something that we can observe? So actually, I mentioned that this is a process that can happen for yeast cells, but it's something that in microcosms, it happens in many situations. For the case of Corynebacterium ammoniaceus, which is a species of bacteria, when we grow them in urea, we have the same kind of process. So urea is a complex carbon source, and the Corynebacterium is enzymatically digesting it. And we can see that by lowering the initial amount of cells. So here we have different time courses in which we see the initial exponential growth of the population, and this is going down as we reduce the initial number of cells in the system. This is from experiments that I was doing a few years ago. And this, again, not the only system in which it happens. Here we have 3 other species that we recently used in an experiment for a paper. And we have here 4 different monocultures, each of them grown at different initial populations. This is an example of logistic growth for Cryptococcus. But then we have 2 Staphylococcus that have what is called a weak allele effect and a strong allele effect for Moraxella. What is the difference between having what we call a strong allele effect and a weak allele effect? Is just the value of this critical threshold. So for a strong allele effect, what we have is that the critical abundance is larger than 0. And for a weak allele effect, this is lower than 0. So the form that this will take for a weak allele effect is something like this. So it means that at low population abundances, the population does not go to extinction. It has a positive growth rate. But as the population is increasing in number, this growth rate goes up. The per capita growth rate goes up. This is what we call the alley effect. And if we continue growing, of course, they cannot grow forever. And then we hit the saturation term, and then it goes down. And then whenever this critical threshold, as I was saying, is negative, then we have a weak alley effect. Otherwise, we will have a strong alley effect. So far we've seen how populations can grow, how this growth saturates as the population abundance is increasing, and also how exponential growth takes different forms if these populations cooperate to grow. Akshit, question?
That's a silly question. So here in the strong alley effect, I agree that stream curve is going down eventually below zero. Right, but like if you are drawing over there in the weak alley effect that you're showing over here, it's not like. Is it basically obvious that when you extrapolate this curve, there's no population density for which you expect to see negative growth rate? Because right now, the x-axis is some 10 to the 4 or something like that. Right?
You're right.
Yeah.
We are plotting the results after 10 to the 4 colony forming units, which is this CFU per milliliter. And this is quite a low density of cells, at which lower than the usual initial density that we use for our experiments. It could be that we go even lower and then the growth rate is negative. Yes, we didn't check, but also it's because of experimental errors. If you go very low, you cannot hit, for example, 100 cells and be sure that you are making the...
Right, so in the experimentally relevant range population sizes, even there, if you basically have positive growth rates every time, then it's a weak alley effect versus a strong alley effect. Is that right?
That's right, yeah.
Thanks.
This kind of crash of populations can happen both for weak and strong. The crash of populations that happens above a certain harvesting rate. That can happen for both cases.
The crash of the population, correct, yes, exactly. So if you mean if we harvest the population, correct, yes. No matter whether the weak alley effect, so whether the alley threshold is negative, this is still a quadratic form, so it's still like this. And as you increase the death rate, you will certainly find a fold bifurcation anyway.
But it cannot happen for the logistic growth.
It does not happen for logistic growth. No.
Another question.
Yes.
The mechanism for the weak alley effect. In these cases, is it the one that you mentioned? This excretion of enzyme,
correct,
collective excretion of enzyme to break down a nutrient?
So yes, and these could be depending, for example, on the initial concentration of sucrose, right? So if the initial concentration is very high, then maybe even with low efficiencies of digestion, then the one cell can still survive and start growing. But if resources are a bit more rare, then the effective of the digestion goes down, and then the one cell will not be able to capture enough resources to have a net growth rate that is positive.
So these were all grown in sucrose environment?
No, these are different. For these ones, we don't really know the mechanism. This was just to show you why we are using sometimes these kind of models, because this is a phenomenological model that captures cooperative growth, no matter whether you know the mechanism or we don't know the mechanism.
So in this case, we don't know the mechanism.
In this one, we don't know. However, we will see tomorrow that we found an ingredient that you can add, and then it softened the alley effect of the 3 species. But still, it's not clear what the mechanism is. So it's some metabolic property that when they found a specific nutrient, it doesn't completely annihilate the alley effect, but it's often seen. Yeah.
So presumably the strong alley effect with negative per capita growth rate at the limit of a single cell means that this is not the correct ecosystem or the nutrient conditions in which this population is growing, that this is sort of an artificial system in which this is being tested, because otherwise how would they ever make populations?
I mean, we can certainly think about ways of doing it in which a population, there is a threshold in the initial population to invade a new ecosystem, for example. And this will happen, for example, for sexual populations. If you only have one individual, that individual will die. But you need at least two.
Two. So yeah. Two, I mean, one or two. I mean, so small number, so one or two, okay. But if it's like the green one,
if it's
then this is somehow saying that this is not the condition of, say, monoculture or correct nutrient or whatever.
Correct. Yes. Yeah. Okay. I agree. Yeah. And it might depend on just other factors, whether there's another species that could help. So here we are moving now into interactions between different species, because actually microbes interact in very different ways. This is microscopy picture in which we have a fungus and bacteria from different species. Is a picture that a colleague of mine, Will Lopez at MIT, took himself with electron microscopy. And actually, this is just to show that bacteria in nature or even in the lab when we have different species, they can interact in many different ways. One way that we have been seeing is this resource competition, which happens when both species are dependent on the same resource. But then there are other ways. In microbes can also try to kill each other by secreting some toxins. That way they free space for them to grow, and they free space from the competitors. But they can also cross-feed each other. So the metabolism that they use to grow happens to secrete some things, and these are resources that are not good for that species, but maybe other species can benefit from it, and they can grow on it. And they can also cross-protect each other. For example, you can have a bacteria that is susceptible to an antibiotics, but you can have a second species that is resistant to antibiotics. And the way to be resistant could be to secrete something that deactivates the antibiotic. If we have this, one species could be effectively protecting not only itself, but also the other one, because it's lowering down the concentration of antibiotic that was killing the first species. Okay, so there are many ways in which microbes interact. And we have started with the most common one, which is competition for resources. And Akshit has introduced a model for that. But yes? Who has a? Yeah? Okay, good. So we've seen this morning how we can model competition for resources. And actually, one pioneer experiment was done by Georgy Gauss, a Russian experimentalist in the early 20th century. And he put two paramecium to compete together in a test tube. So parameciums are species that unicellular eukaryotes that can feed on smaller microbes like yeast or bacteria. And what he had is a medium with bacteria, and he put the two species inside, or just one. So we have the paramecium aurelia and caudatum, and we see that both of them can grow in this medium where they are separate, and where we have only one species. But when he put the two of them together, he shows that caudatum was going to extinction. And Georgy Gauss coined this principle of competitive exclusion. But we know that competition doesn't need to be leading to exclusion. It happens when they are competing exactly for the same resources, but we could have that the two of them can live out of different resources, but still be competing from space in the ecosystem. In that case, there could be a situation where they both find some space and they are feeding on something else, so they compete. They will be better if the other one was not there, but still they can stably survive in the system. Could we? Yeah. Actually, what Georgi Gauss found was a case in which one species will dominate the other, or it will exclude the other by the principle of competitive exclusion. And then no matter at which initial abundance you start, this species will take over the system and the other one will go to extinction. One way in which we can plot the trajectory of the system is by thinking not only about the abundance of one species, but about the fraction of the species. So this is how much I find of species one relative to the total amount of individuals in the system from both species. So exclusion is not the only way we can, not the only outcome we see out of experiments. We can also see coexistence. This will be a case in which if we start with a lower fraction, initial fraction of one species, this will grow. But if we start with a high fraction of that species, it will lower down and it will reach a stable abundance or a stable fraction. And in this case, none of the species are at 0 or 1, but both of them have some positive abundance, and they reach this stable state. And there is an alternative canonical outcome, which is bistability. And in this case, the outcome depends on the initial fraction of the species. So if one species starts at a high abundance, high relative abundance, then this species will grow at the expense of the other and it will take over the system. The other species goes extinct. But if we start at a low abundance of that species, then the other competitor is growing while this one will decay to extinction. And this is what we call bistability in pairwise competition, so in the competition between two species. These are three canonical outcomes. I will say 90% of the time that we do an experiment in the lab in which we put two species together, we're going to find one of these three outcomes. Sometimes you might find a different thing. You can have a population that is a community that is oscillating, and or you can have a bistability in which one of the stable states has some coexistence instead of going fully to 0 or fully to 1. But those are rare cases. The majority of them will end up in one of these three situations. So we would like to model at least these three generic cases. And for this, what we can think about is to construct a simple model. We have seen already how populations grow logistically, and we are going to add an interaction term for the competition between the two species. And the model I'm going to write is the Lotka-Volterra model that Stefano was presenting yesterday for many species, and I'm going to do the case in which we only have two species. Let me. Okay. So here we recover the notation that that Stefano wrote yesterday, in which the abundance for the species I was proportional to 1 my EIG. Maybe I'll write it explicitly with the. All right. So here again, we have for species one, a logistic growth, and we add a term for the interaction of the second species. I started by writing it this way, because this way, intuitively, we can think about that K1 is the carrying capacity of the system. So when the population X1 reaches the abundance K1, then growth goes to 0. So we can have a stable population of K1 individuals, if you want, in of X1. But if we have the second species, this term here measures how much space is the second species occupying relative to the space that you need. Okay, so somehow, if this beta is larger than 1, it means that the competition with the competitor is stronger than the competition with the individuals of the same species. Okay, so no, not necessarily. Yeah. And now let me do some renormalization, because it will help with the analysis later. for this. So if we change x by y and just renormalizing by their carrying capacity, now y will take values between 0 and 1, and then b will be the strength of competition in this new way of measuring the abundances. Okay, so if we do this, we can capture the different outcomes of competition in the log of this beta to 1 and log of this beta 1 to b1 2. So in this case, we find that whenever the two coefficients b1 2 and b2 1 are larger than 1, we will find bistability. This is and then here we will have species 2 wins. We find coexistence here. And species 1 wins. Here I'm going to give a little bit of time, few minutes to just play in computing the null clients. As Akshit had mentioned, these null clients are the lines or the curves where dx1 x2 is 0, and actually I want to play with the normalized version. So let's find the null clients and try to use them to find the fixed points. And later on, we will see how this leads to the different competition outcomes for pairwise interactions. Okay, so what are the null clines for this? There are two that are quite obvious. Mm-hmm. Mm-hmm. And also y can be 0, y1 can be 0, or y2 can be 0, right? So we have that we have the the vertical axis and the horizontal axis are both null clines. And then we have, sorry, y. And then we have that y1 is equal to 1 minus alpha 1, 2. And the fourth null cline is y2 is 1 minus alpha 1, y1. So by combining all of this, how many fixed points can we find? 4, that's right. One of it will be 0, 0, right? The other one is at the carrying capacity that in the normalized version is 1. Okay, and we can also have only the other species. And then there is a fourth one where these two null lines cross. Okay, so for this one we will have 1 minus sorry, yeah, I'm very used to call these B's alphas, and you will see that this is the case also in other papers. So if I write alpha again, please just tell me. Sorry. Okay, and if we do the maths, here is where we will find the other fixed point. So these 3 fixed points, so these 3 first fixed points, they always exist. And this one for biological populations, it only makes sense if it is in the positive quadrant. Okay, so both this term and this term has to be larger than 0 or equal or larger than 0. There are no negative populations. Okay, so then if we play with the values of P in the normalized version, what we'll see is that in this case, we say that this is a null cline and this is also a null cline. The other 2 null clines take this form. It's the inverse of beta 1, 2. And here, it will be the inverse of beta 2, 1. So remember, what's the meaning of this null line? This one is the one for which the growth of x1 is equal to 0. Okay, so when we are exactly at this null line, the trajectory of the system, so if there's the population is here, we have this amount of x1, this amount of x2, x1 here is not going to grow. And if we are here, x2 is not going to grow. What does this mean? So we know that for everything that is below this line is for x1 to grow. So everything that is below this line goes right. And everything that is above this line will go left. So by this means that if we are here with some amount of x1 and some amount of x2, x1 will grow. It will go to the right. But if we are above this, it's like being above the carrying capacity, we will go to the left. Is this clear? Yeah? Okay. Any question about that? Now, what happens to the second species? So we know that anything below here is for this species to go up, and anything above here for this species to go down. So if the community starts here, x2 will go down, okay? So another way of seeing this is that anything that crosses this null line can only go up because x1 is not moving, is not growing. Okay, so here we have only trajectories that go up. And anything that goes here cannot go up or down, only left or right. But we are above this line for which we have that x1 is dying. So anything above this line can only go to the left. And just by this analysis, we can now understand whether the different fixed points of the system are stable or unstable. Okay, so if we are nearby here, we will go up or down, but we will always be pushed to the left, to the extinction of x1. And then this species wins. So we have a stable point here. We already knew that the 0, 0 is unstable. If you have a little bit of the population, this will grow. And then we have this other stable state, which for x1 is stable, but as soon as we put a little bit of x2, we go away from it because x2 will start growing. So then this one is also an unstable point. So with this graphical analysis, we can know that whenever this interaction term is lower than 1 for species 1, larger than 1 for species 2, we will have that species 2 always win. Okay? I'm going to quickly draw the same analysis for the other ones, and then we will move on. So, so in this case, this one here, this one here, and I leave it as an exercise to see that then these two are stable, and this one, which is the fourth fixed point that now has a positive value on both axes, this will be an unstable point. Okay, so the population here moves either here or here. Mm. Um, okay, sorry. Yes. Okay. Yep. I have, yes. In, sorry, say it again, in this. Yes, and I got confused about that at some point with the colors and the things, but
yeah, I think in the base table case you're just missing yeah, the solid line should be.
This should be a solid line, right?
I think the other one.
That you are coexistent, you have the solid line, the dash line.
Right. Okay. This is. I was getting confused about that. So. So the lines were okay, but the dash lines are not. Mm. Right. So this. Right. So we are with something like this, right? And then in here, you would like to have this one as a solid line. Are we all good now? No? Which? Okay, again. Right. No, it should be good, right? Yeah. Okay. Good. So if it's still not all right, let me know, and we can check on that later. But the way in which the four outcomes come is plotted all right. I want to just show you how we do these experiments in the lab, where we just put the two species together. And a simple way to do it is to get two species. We have now a Corynebacterium and a Lactobacillus. And then you put them at a low density at the beginning of the day, and we let them grow. These grow to saturation. And then the next day, it keeps growing. You dilute them to renew the resources, and they can continue to grow and get saturation again. And we do this day after day. And during the experiments, these species are growing and interacting, and the relative abundances can change. So when we did this experiment for these two species, what we observed is a case of bistability. So we have here that one species can take over the other one if the initial fraction or the initial relative abundance is a threshold. But if the orange species starts at a lower fraction below this threshold, then the green species will take over that one. So we will have basically that the competitive interaction from the green is able to exclude the orange microbe. To demonstrate that this was a stable state, we actually exposed them to some amount of migration. So every day we will include a little bit of fresh cells from both cells in the system. And despite we are doing this, the outcome will not change. Will still be bistability. And somehow this immigration can also represent a bit what can happen in many ecosystems. For example, in the gut microbiota, we have a community of many different bacterial species and microbial species. And every time we are eating, some fresh cells coming from the outside world will have a chance to get into the system as well, right? So with this kind of system, we have now a bistable system where we can play to we can study the effects of perturbations that could drive the system from one state to the other. Why is this relevant? Because there are situations in nature in which perturbations can drive dramatic shifts between the states of different communities. One example is the one about antibiotics. We take them usually to fight pathogens, to fight an infection, but actually in the gut microbiota, again, we have many different species that, as a side effect of the treatment, will be affected by the antibiotics. So this community that we have in the gut usually have a moderate to high diversity. And when we expose them to an antibiotic, during the treatment, some of these bacteria can suffer and can die. And then this diversity, which means how many species we have in the system, can go down. The hope is that after the treatment, we will recover the initial state of the community. But this is not always the case. In some cases, they've seen that after an individual has been treated with antibiotics, the treatment is ended and some months are passed, and the individual has still not recovered the diversity of the species that it had before. So it is very difficult to understand the mechanisms of these transitions. Here we have a very complex system in which the lifestyle of the person and the immune system plays a role. The kind of antibiotic also plays a role, but also evolution of antibiotic resistance by the microbes could also play a different role, as well as many others. So we would like to be able to understand at least the basics. What will happen if I have like two species that are in competition and they happen to be exposed to some antibiotics, okay? So when we when we think about what is the common approach in medicine, is that if there is a particular pathogen that you want to fight, and this pathogen is sensitive to an antibiotic, you just give the treatment and you expect that it's going to be effective. So based on that, you will expect that the more sensitive a species is, the more likely it will suffer from an antibiotic perturbation, even when they are in community. So to try to approach this question, what we did is to measure the antibiotic susceptibility of our two species to different antibiotics. Here I'm showing the case for chloramphenicol. So what we do is we grow separately each species at different concentrations of the antibiotic. And we see that the growth rate of that species goes down, or the growth at saturation goes down, and it goes down at a lower concentration for the green one than for the orange one. The measure we are taking, or we are using here, is the IC50, which is the half maximum inhibitory concentration. Okay, at what concentration I'm reducing the growth to 50% of the original one. So based on this, we conclude that the green bacteria is more susceptible than the orange bacteria. And we will expect that then we can use this antibiotic to induce a switch between the stable state of the competition that is dominated by the green microbe towards the orange microbe. But we did this experiment, and this is not what happened. We exposed to chloramphenicol for 24 hours, and the community responds to the perturbation, but after we remove the antibiotic, the green microbe was able to recover and take the community again. And perhaps more surprisingly is what happened in the other case. So here we have the orange microbe dominating the system. We are in the alternative stable state of the community. We expose them to chloramphenicol, and after the antibiotic, during the recovery process after the treatment, we see that the green microbe takes the opportunity and takes over the system. So even if it's more susceptible to the antibiotic, when in competition, it can still benefit from the perturbation.
In this case, even before the antibiotic was administered, there were changes in the relative population.
Yes.
So,
if I look at both these curves, which you've shown here, even before the pink bar, which is where the chloramphenicol exposure is there, there is you're not in a stable state before that. You may not be in a stable state. And in particular, one of them is dying and the other one is growing in the top one. And in the bottom one of them is sort of stable and the other one is dead.
In the,
I mean, it's sort of very low.
Correct. Yes, that's right. So it's below the detection limit. That's right. We know that there is there because, as I was mentioning, we did this experiment in the presence of some immigration as well. So every day, in addition to the microbes that come from the previous day, there are a few fresh cells from both species. So somehow in our system, there will be no complete extinction, but given the bistability, one of the species will always be pushed down to low abundances.
Right.
Yeah.
Okay.
And your question is about the fluctuations?
I also wanted to know what is the level of chloramphenicol? Is it between this 3.1 and these?
Yes. So here, I think in this case we were around here. So we will take concentrations that will not kill at least one of the species, but this is not very, very important because it was not a result that happened only for chloramphenicol, but for different antibiotics. So we tried 8 different antibiotics with 8 different mechanisms of action. Well, with different mechanisms of action, and here we have ordered by the relative difference or the ratio of the susceptibility. So for the first antibiotic, both species are more or less equally susceptible, and for these ones here, the orange one is almost resistant and the other one is very, very susceptible. So for all of these antibiotics, we have more sensitivity in the case of the green one, but still, up to here, the green one was always benefiting from the perturbation. Then we observe that for these 2 cases, we have no changes of state. If we start with green, we end up with green. We start with orange, we end up with orange. And then only when the susceptibility ratio was very, very different, then we can use these antibiotics to favor the orange microbe. Would that address your question, or tell me if there's something that
I'll have to think about it.
Okay.
Yeah, but I think this does address partly that question. Yeah.
Okay. Yeah, and the perturbation and the fluctuations are something that are kind of common. So here we have a log scale, and still there are so strong interactions that in the end, experimentally, with the experimental errors, we get some of these fluctuations. But if we continue the experiment without the perturbation, this will not change the state. It will always get the green one at a high abundance and the other one at low abundance here, and vice versa.
So one thing which is puzzling in the second case,
yes,
is that when you add the antibiotic, at that time the orange one is declining and the green one is rising.
Yeah,
and that I mean, does not seem to be consistent with the first curve there.
Correct.
Unless there are some interactions which are going on which we don't know anything
Correct.
about.
Yeah, I agree completely. So this is the combination of the antibiotic exposure and the competition with the other. And the competition with the other has suddenly dropped, which removes a little bit of pressure. And we are still at a concentration that is not killing the 2 species, and we are applying migration. So the net effect was that this led to a significant increase in the population during the exposure. But it's because of the combined effect with the competition. So actually, to understand these kind of transitions, we used a modification of the Lotka-Volterra model. So here, these alphas are the BIJs that we were talking about before. And we modified the model to take into account the experimental conditions. So we have some immigration, which we call dispersal. So this is a constant addition of cells into the system. And then we have the effects of the antibiotic. This is a death rate that is different for each species, the first species and the second species. And we have this death rate only during a window of time, the window of time of the antibiotic treatment. So if we take into account again that we have So if everything, if the 2 species were the same, then most likely everything will be about the death rate, the antibiotic death rate. But here we have different parameters in the model. And of course, one that comes to attention is just that they don't need to be growing at the same speed. And it happened that the green species was growing faster in the absence of antibiotic than the orange microbe. Okay, so now we have a combination of interactions that we know that they are strong because this system is bistable, and we have different growth rates in the absence of antibiotics. So this one is the one that has a higher death rate associated to the antibiotic, is more susceptible, but it's also one that can grow faster when there is no antibiotic. Okay, so we made some simulations about what we could find, and we see that if everything was the same, so if they had the same growth rates, then everything will be intuitive. We apply the death rate, and then we can have a switch towards the orange microbe winning. But the moment that we have this trade-off between sensitivity and growth rate, we can have situations like the ones that we observed experimentally. So the more sensitive species will still benefit from the perturbation. And we can actually draw the phase diagram for how these transitions happen. And we could draw them on the axis of the ratio of the two death rates. So here we fix the death rate of the first species, and we decrease the other one. Here we fix the growth rate of the first species, and we decrease the one of the other one. And then we apply these antibiotic perturbations. And what we see is that there is a region in which the green microbe always wins, another one in which both the states are resilient, and another one in which the antibiotic perturbation would favor the orange microbe. And actually, this recapitulates what we observed experimentally, that for a given ratio of our two species, as we increase the susceptibility, or as we decrease the ratio of these death rates, favoring the slow grower, which is the second species, then we will cross the different phases in a particular order. So we will have first antibiotics for which the green microbe wins, then some other antibiotics for which no one benefits, and then some other antibiotics for which we have the orange microbe winning the competition. And experimentally, we were then able to play with the growth rates of these two species. And when we make the orange microbe a faster grower, then you can use the same perturbations, and they will always favor the orange microbe. So we did this just to show that everything was dependent on the growth rates in the absence of antibiotics. So far, I've talked about the growth rates, sorry, the perturbations that were temporary in time. And this is one kind of perturbation, which are called pulse perturbation in ecology. But a different perturbation that you can have is one that lasts over time. So this is what they call a press perturbation. And this usually is associated to a permanent or a long-term change in the environmental conditions. One way to do this is to, one way to model this is to include a death rate in our model. And this is what some colleagues did a few years ago. I'm looking for, yes. So they modeled the addition of a mortality rate or a death rate into experimental competitions between pairs of microbes. So they did, when they first did the modeling, they saw that if we add here a death rate that it's the same for the two, and yes. So to model the added mortality, they just modified the Lotka-Volterra model to have this form in which both the species are now exposed to an external death rate. And this is captured by this parameter delta, okay? What they found, I don't have the time to do the algebra now, but basically what they found is that you can, again, do a different kind of renormalization, and then you will find renormalized variables, let's call them Z instead of I, in which you will have again the same model with this form without a death rate. And this death rate will be incorporated into the competition terms together with the growth rate. And if you're curious about that, we can show tomorrow how they renormalize the model. But basically, what they found now is that here, where we have our phase space of the log of the interaction terms, now renormalized for this kind of death rate, what they found is that as they increase the death rate, this will go straight with an angle of minus 45 degrees in this direction. So by adding a death rate, you could cross for a given pair of species from a situation in which one of them wins. They have the second species here, which was the slower grower. And then it will be favoring the fast grower, which is the first species in the theoretical model. So here, just by adding the death rate, they predicted that they will cross first a region in which the system is bistable, and then it will go to the fast grower of this bacteria winning. But for a different pair of bacteria, maybe the interactions are more over here, and as they increase the death rate, this will cross the different outcomes in this way, going through coexistence. So actually, that's what they did experimentally. Here, you have a Pseudomonas against an Urantiaca species. And under these conditions, which are with zero death rate, zero added death rate, we will have that the Pseudomonas will win. And then they increase the death rate, and they will see that while before we have the exclusion of these species, now we have the coexistence of the two. So here is the fraction of the two species over time. And here are the experimental results when you keep increasing the death rate up to 0.3 by hour. And then in this case, you will have that the second species wins the competition. So somehow, the Lotka-Volterra model and extensions of the Lotka-Volterra model can capture situations of experimental competition. This is a case in which the pair of species was crossing through a different region. So in this case, the community becomes bistable. So either one wins if it starts at a high abundance, or it loses the competition if it starts at a low abundance. And then if they keep increasing the death rate, then they will have them go into the region where the first species wins. And then just to finish, okay, this was inconvenient, but basically what I wanted to show is that with the same model, they later analyzed what happens when they increase the temperature, and the temperature actually has some effects that we can model on the growth rates of the species. So what the authors found is that while increasing a death rate will push the competition in this direction, increasing the temperature will go the other way around. So this was favoring the species that grows faster, but increasing the temperature will be favoring the species that grows slower. And this is because of how the relative changes happen as you increase, the relative changes in the growth rate happen as you increase this temperature. Yes.
I just continue. In the earlier case, where they were all going down this way, did you say that the perturbation was such that the delta term was the same for both the species?
Yes, in that case, yes. And it doesn't have to be the case. If you change the death rate so that it's higher for one than the other, then you will change the angle of how this eventually favors the fast grower.
Okay, so the difference between this perturbation in which you're applying an antibiotic, which reduces the death rate of both of them, how is it different from the earlier experiment in which which you reported before this?
It will be not very different if we let the species live in antibiotics over time, but it was we just did it for a lapse of time, as you do with antibiotic treatment. Treat it with some time, then you release the pressure and you let the community to recover. So while we apply it, it could be that they also cross this way, but then when you remove it, you should be coming back to.
I see. So in the earlier experiment, you were letting it come back. In these experiments, you just
yes,
keep applying the antibiotic persistently.
Correct. Yes.
I see.
Mm-hmm. And then the last thing I wanted to mention is that in many different works, the Lotka-Volterra model has captured quite well what happens for competition between pair of species in the lab with microbes, and then another bunch of work that I don't have time to go into has been looking at how we can predict the dynamics of larger communities. So this was for 2 species, what happened when we have more species, and Stefano is giving 4 lectures about the modeling of more complex communities. But just to show that some of the phenomenon that we study for 2 species can be then extended to provide some understanding of some patterns that can happen in more complex communities. So here I'm just mentioning that some of the same authors that were working in how death rate versus temperature affect the competition between 2 species, then use the results to predict how the composition of a species in natural microbiotas, concretely in the ocean, will change for different temperatures. So they will just looking at data sets for microbiota at different latitudes, and then they will see that at warmer latitudes in the center, you will have a higher proportion of slow growers relative to the ones that you have at higher temperature. Correct. And the way they look at this is just by looking at the genome and having some genes that are associated to being a fast grower or a slow grower. So with this, I'm already out of time. So I just wanted to summarize that we've seen that cooperative growth, which is called also the allele effect, can lead to bistability in populations. The 2-species model captured the 3 canonical outcomes of pairwise competition. We have bistability, coexistence, and competitive exclusion. And we can use modifications of this model to predict how communities respond to different perturbations. And on the way, I've shown you how working with experimental communities in the lab with microbes can be used to have a feedback between the development of theory and the testing of some of the predictions. So with this, just thank you for your attention. If you have questions, I'm happy to take it.
In one case, you are explaining that when there are 2 species and you applied the antibiotic for a very small amount of time, so one was dropping and another was getting up. As said, sir said, it was not it was contradicting itself, because one was increasing while we were applying that small amount of antibiotics. So is that cross protection. Yeah, this one. Yeah. Is that cross protection that we call? And that the second one?
It could be cross protection, but like if you see data like this, it could be cross protection. We checked in separate experiment, and it was not cross protection. The way you will check is you grow microbes of one species in one under the antibiotic, then you remove the cells by filtering, and you put the other species, and you will see that it still dies. If it doesn't die, then it's cross protection somehow. This is a way of testing. It could be still another form of cross protection that we cannot detect with these experiments. But basically, my sense is that this was a combination of the antibiotic effect and the competition, the interaction between the two species. So we know that the interaction is proportional to the abundance of the other species. And here, the abundance of these species just went down. So you were releasing competitive pressure, and maybe this allowed for the significant growth that we see here.
So when we like to see this calculation on this graph, we isolate each of the species, then we apply this, right?
Where?
On the
here?
first, yeah.
Yes, here I'm putting the two of them together, but correct. So here it was just the green one or just the orange one. And community, so the competition was happening only there.
So in the model that you had for the same system, I think there was a death term that was already there. So basically here, the T for which you apply the antibiotic here is a very small pulse.
Yes. Yeah.
Okay.
So if it will be actually, so it's not a delta. It's not a Dirac delta.
Right. Okay.
It's 24 hours.
What I mean to say is the T interval here is very small. Because otherwise it's the same as the press model that you were.
Yeah. Correct. Okay. Yeah, exactly. So here, this is what we call a pulse perturbation. If we make this forever, we extend it forever, it becomes a press perturbation in which the system has now a different environment where this is.
Technically, it's the same model.
Yes, I agree. With the exception of the migration that was not incorporated in the other paper. Anything else? All right. I think we can call it for lunch. So thank you again.